Okay, so now we are switching to single-cell RNA-seq and having Nathan Castro Pacheco presenting his work on the STARsolo-Quant analysis. Thank you.
Thank you for the introduction. I just want to thank you all for the opportunity. Thanks for staying late. I know it's not really easy to stay late. I'm sure after a long day you all are tired. And I'm going to be talking a little about the quantification of relevant transcript isoform abundances from single-cell RNA-seq data and my involvement in the creation of STARsolo-Quant, which is a tool that's part of STAR that's trying to estimate the relative abundance of different transcript isoforms. So one of the primary ways single-cell RNA-seq data is being collected right now is through droplet-based methods. And there's been many droplet-based methods developed over the last few years, but they have some general characteristics which they share. The main one being the fact that the reads generated are barcoded by cell barcode and unique molecular identifiers. I'm sure a lot of you are familiar with this, but I'm just running through this to make sure everyone is clear about what these are, because they're important for the model. So cell barcodes are reads that or like short sequences at the front of reads that associate the read with a given cell, and unique molecular identifiers are also short sequences that associate each read with a particular molecule that was present in the cell prior to amplification. In addition to these barcoding characteristics, these methods are very high throughput in their capacity, which means they've led to the identification of many new cell types over the years, but that's been at the expense of relatively high dropout rates, which means you don't really have that many reads per cell. And that can be relatively problematic, especially if you're trying to do something like quantify relative abundance of transcript isoforms. So just to be clear, a transcript isoform can generally be defined as a structurally distinct RNA molecule produced from the same gene as a result of different pre- or post-transcriptional regulation mechanisms. A couple of those pre- or post-transcriptional regulation mechanisms are alternative splicing, alternative transcript initiation, and alternative polyadenylation. Alternative polyadenylation is characterized by the addition of a poly A tail here at the 3' end of the transcript. As you can see, these 3 transcripts at the beginning have the same polyadenylation site, and these other transcripts have distinct polyadenylation sites, which obviously gives them distinct structure and function as a result. So the majority of studies to date have been focused primarily on gene expression or transcript isoform expression, not on a single cell level. So while those studies have been very useful in sort of defining what the gene expression profiles look like for different cell types, you could imagine scenarios where we might want to sort of quantify the transcript isoform expressions for different individual cells, which might have, per se, similar patterns of gene expression, but different transcript expression profiles. So an alternative to the conventional approach of gene expression studies is transcript compatibility accounts. Rob Patro mentioned in his talk the idea of equivalency classes, which is relevant to this, and I'll get into detail about that if I have time. And so after considering all those different aspects, the question remains of whether or not we can quantify transcript abundances from this droplet-based single-cell RNA-seq data. So that brings us to further detail about the cell barcoding feature of single-cell RNA-seq data. So very generally, after the cells are lysed within these droplets, the transcripts represented by these black lines are hybridized with the unique molecular identifiers that are attached to a microbead. And after processing, you end up with this library of relatively short reads. Each read has a cell barcode and a unique molecular identifier. Just to reiterate, the cell barcode associates the read with the given cell, and the unique molecular identifier associates the read with a particular transcript that was present within the cell at time of sequencing. So for sake of example, let's say this transcript that's associated with this orange UMI was this particular, or was a copy of this particular transcript. As you can see, reads or the cDNA sequences map to distinct regions of the transcript occasionally, but there's also the case where these reads that map are multi-mapping. So that means that the transcript that this unique molecular identifier was attached to, the identity of it can't be resolved, and that's where the main problem arises when you're trying to do transcript isoform quantification from single-cell RNA-seq data. In cases where you do have a read that maps uniquely to the unique region of a transcript, that's not a problem, and then you can associate that particular UMI with a given transcript, but you can imagine that's not typically the case. So you can use sort of a key property of single-cell RNA-seq data or droplet single-cell RNA-seq data, which is this fact that reverse transcription begins from the 3' end. And that generates a 3' bias in the data or in the reads from the distance of the transcript termination site. So you have a much higher likelihood of reads being generated from closer to the 3' end as opposed to far away from the 3' end. And we use that to sort of resolve reads that map ambiguously to multiple transcripts, and I'll detail exactly how we do that in a bit. But as you can see, compared to bulk RNA-seq reads, which have very low densities at either the 3' or 5' ends, and sort of, I guess you could call this somewhat uniform distribution from between the 3' and 5' end, this model is this distribution is very distinct, so it allows us to take a distinct approach as opposed to bulk RNA-seq data. So just to further reiterate the point that because single-cell or droplet-based methods are relatively high throughput, that leads to a low read depth per cell. And in order to circumvent that problem that comes with these relatively low number of reads per cell, you can group or cluster these cells by their gene expression profiles and then take all the reads within these clusters when you're trying to quantify transcript isoform abundances. And that's because if you have a relatively low number of reads, there's really no way to accurately quantify transcript isoforms for individual cells. And that's just going to be the result of the fact that we don't have that many reads per individual cell with the current technologies that we have. So our approach after forming these clusters using existing clustering tools, which I'll detail further in a bit, is to take all the uniquely mapping reads and add them to this distribution, which we call D of D. And D of D is this distribution of distance from the transcript termination site for the reads, but it's obviously going to be unique between data sets. So we generate this distribution D of D for every single data set that is inputted. And then for the reads or the UMIs that can't be solved or as a result of uniquely mapping reads, we'll apply this expectation maximization approach in an attempt to resolve those and assign a probability of UMI or a transcript being associated with a given UMI in the cell of origin, right? So you have this likelihood expression, which is given theta, right? If you have theta given U, where capital U represents all the UMIs within a given cluster, and theta represents the isoform, the relative isoform abundances of different transcripts, you can take the product of all of the D of Ds for all the reads in a given UMI and multiply that by the theta, summing over the transcripts, and then finding the product between all UMIs, right? And that'll give you a likelihood value for each one of the thetas, and then you can maximize those likelihoods using an expectation maximization algorithm, which I won't get into the details of for sake of time as well. So on that note, this is a more graphical representation of the workflow for STARsolo-Quant. It begins by using STARsolo to map reads to a reference genome and then quantify gene expression. And after outputting that gene count matrix, after quantifying gene expression, you can cluster cells using existing tools like SARAT or ScanPy. And then after getting those clusters, you can group the reads from individual cells based off of those clusters and then perform expectation maximization on each one of those individual clusters. And it's worth noting here as well that we are considering the idea of clustering by transcript compatibility counts as opposed to gene expression, but for this presentation, we clustered by gene expression for the data that's generated. So the benchmarking we began with was relatively basic. It only represented very small simulation sets, and it was just to show the importance of UMIs as well as reads, which I'll show you in a bit. With unique molecular identifiers being associated with unique molecules being present in the cell prior to amplification, you can only imagine that if you have a very low number of unique UMIs, your estimations are going to be fairly limited in their specificity, right? So the ground truth I should have mentioned previously is this ratio of 0.3 to 0.5 to 0.2 of these colored transcripts. And as you increase the total number of UMIs available in the simulation, it becomes increasingly more accurate. After this certain point, it doesn't really matter if you add more UMIs, and that's because the ratios are relatively simple. So the importance of read count is more interesting, I guess, I would argue, I would say. And that's because when you have relatively low number of reads, you can imagine that they would map to this region that's shared between these different transcript isoforms, and each read will carry more sort of influence over the estimations that are made. So in very low read counts, you get estimations for these red and gray transcripts which aren't even present in the ground truth. But as you increase the total number of reads, you get closer and closer to the ground truth with these error bars representing plus or minus 1 standard deviation decreasing substantially over time or over total reads. And we think that's the result of having increased number of reads. You increase the chance of having transcripts or having reads which map to the transcript unique sequences of these transcripts. And lastly, in terms of simple benchmarking, we also tried to quantify isoforms with identical transcript termination sites, and this did cause a little bit more problems, right? So as previously mentioned, if you have a multi-mapping read, it's typically resolved, or it's attempted to be resolved by comparing the distance from transcript termination site between the transcripts. But you could imagine if you have identical transcript termination sites, the model is going to have a very hard time predicting the relative abundance of these transcripts. And in order to get around that problem, you can also note that these transcripts are very structurally similar and have only a very small unique sequence at the 5' end, which you won't get many reads at because of the 3' bias. But in order to solve this problem for now, what we do is we aggregate transcripts with very close transcript termination sites. So as opposed to generating individual estimations for relative transcript isoform abundance for each one of these transcripts, we're just proposing sort of aggregating all of those relative abundances so you have knowledge that it belonged to one of these 3 transcripts. So for our larger scale simulations, we used Splatter to generate a gene count matrix and then inputted that gene count matrix into Minnow in order to generate the number of UMIs per transcripts as well as the number of reads per UMI. We had to replace the cDNA sequences generated just because they didn't fit that 3' distribution that I previously mentioned. But upon assessing the simulated data, we got a relatively strong correlation between the true UMIs per transcripts and the calculated UMIs per transcripts. And we got an even stronger correlation when we applied this approach of aggregating transcripts with relatively close transcript termination sites. It's worth noting as well that there are a significant number of transcripts on this left side for which the true UMI per transcript was 0 and the calculated UMI per transcript was calculated to be more than 0. And that is the result, or you could argue, or I'm starting to think that that's the result of cases like these where you have relatively low number of reads. So estimations are being assigned to transcripts that share some sequence. So that's a result of not having enough reads. And this is our example in which we applied it to a real data set. And this is the 10x v3 data set, which is, has a good number of cells. All of these clusters, with the exception of clusters 7 and 8, expressed PPP1cc. And these are the isoforms for that gene. And so what we ended up doing was these 2 transcript isoforms have relatively close transcript termination sites. So we sort of grouped their quantification as I was suggesting we would do. We grouped them in this column here. So these are the groups here. And as you can see for T cells and myeloid cells, for the most part, the relative, oh, and it's also worth noting that the reads were grouped by these clusters up here, right? So the reads for individual cells were grouped, as previously mentioned in the method, and then this transcript isoform quantification step was performed. So as you can see, there was not a very significant difference between the usage of these different transcript isoforms and a number of these cell types, but in the B cells and the natural killer cells, this first transcript isoform was suggested to be used a lot more. And there might be some biology behind that, but there's still a lot of analysis and benchmarking for this tool to be done in order to be sure of that. So it was just an interesting example to see, and there's a lot more interesting examples that there wasn't time to show in this short presentation. So on that note, STARsolo-Quant does a good job at circumventing low reads counts per individual cells and solves that via clustering. and it takes into account the characteristics of droplet-based technologies, and it performs relatively well on simulated data sets. And in the future, what we'll do is try and analyze the statistical significance of differential TTS usage by doing what we call pseudo-replications, where we take a cluster, separate it into two, perform expectation maximization for each cluster, and compare them to see sort of the relative effective noise between these. And we also are looking to accelerate the expectation maximization step using one of the methods that Rob Patro mentioned earlier, and also assess the effect of clustering by transcript compatibility counts as opposed to gene expression. And I'd like to acknowledge my lab members. And I first came to Cold Spring Harbor as part of an undergraduate program. So I'd like to acknowledge the people who had that undergraduate program sort of set up. Thank you.
Okay, so questions? Okay, over there.
Thanks for the talk. In the likelihood model that you showed, you are using the distribution of basically the distance from the start site, which drops off quite significantly.
Right.
And in the annotation, there's very poor annotation of alternative 3' polydenylation site. So how do you account for that? Are you just relying on basically having them properly annotated, or do you do anything special with them?
In terms of annotation for the poly A 3' ends, we don't really use those annotations. There's no dependence on those annotations. It's more or less generating, sorry, I'm trying to get back to that distribution. It's more or less dependent on this generation of this distribution that's generated from data set to data set, which doesn't depend at all on these annotations of the poly A tail. That should be something that we could consider in the future, just because there is polydenylation at the 3' end, but as the model currently stands, it doesn't really consider that.
More questions? I have one.
Okay.
So, a study that we did in our lab and we published last year, we were looking at the ability of UMIs to discriminate transcripts in human genes. Mm-hmm. And we found that it was maybe something like only 20 or 30% of the multi-isoform genes could be fully discriminated by UMIs because of the 3' bias, right? Is your analysis also kind of resulting in this kind of numbers? Is your analysis aware of, that the great majority of the genes you cannot discriminate all transcripts?
Right, so you really can't discriminate all transcripts, so it really is more of a probabilistic approach as it stands. So it's not a definite discrimination of all other transcripts. It's what it does as it currently stands is, let's say you have all the reads for a certain UMI that map to, let's say, 3 transcripts that are very structurally similar. If by any chance you have one of these reads that also maps to a transcript or maps uniquely to another transcript, what it will do is it'll prioritize the transcript that it maps uniquely to if it's mapping to those other transcripts as well, because it sort of prioritizes reads that map to the unique regions of these transcript isoforms. And there unfortunately are cases where these unique regions are very small, and that's probably why you experience those types of numbers. But that's currently as the model stands, and there is definitely a lot of room for further analysis, and I'm sure we'll run into situations where that's the case in the future.
Thank you very much.
Mm-hmm.